welcome you all. I'm Dr. M. Leila, Assistant Professor, Department of Zoology, Shishalda College for Women, Salem. I'm going to talk about vectors in biotechnology. So, the DNA that carries the desired gene to the host is called as gene cloning vector. In simple, vector is also a DNA molecule which is used as a vehicle to carry a particular DNA segment into the host cell. So we can tell like the DNA which carries the desired gene to the host cell is called as gene cloning vector. It can be also known as cloning vector, cloning vehicle or carrier DNA. In molecular biology, a vector is a DNA molecule used as a vehicle to transfer foreign genetic material into another cell. It has the ability to self-replicate and integrate into the host cell. Usually, genetic engineering involves the transfer of desired gene into the host cell that has to be manipulated. That desired gene may be hydrolyzed by cellular enzyme immediately after introduction. Further, the chance of its expression is also very poor. So, in order to overcome these problems, the desired gene is inserted into a suitable vector for gene cloning. That is, the DNA of interest is inserted into a specific site of vector and ligated by the enzyme which is called as DNA ligase, and then vector is transferred into host cell for replication. The four major types of vectors are plasmid, spiral vector, cosmic, and artificial chromosomes. And the most widely used in the field of genetic engineering are plasmid and bacteriophage. rDNA technology, which involves the isolation of genetic material from the host cell, cutting the gene at the recognition sites, and the important point here is insertion of vectors. So we already know the importance of vectors in the rDNA technology. So in order to overcome all the problem, we need to select the vector in appropriate manner for amplification of gene copies. So the next step will go smoothly. Next is amplification of gene copies, ligation of DNA molecules, insertion of recombinant DNAs into the host cell. Coming to features of cloning vector, a cloning vector, how the cloning vector should be. A cloning vector should possess an origin of replication for self-replication. So after going inside the host cell, it needs to replicate itself. So it should have a region which is having replication. And the size should be small so that insertion is very easy. Even though it is small, it is capable of inserting large segment of DNA. The plasmid, even it is small, it is capable of inserting large segment of DNA. Should be selected as a cloning vector. It should be capable of working under both prokaryotic and eukaryotic system. It should have restriction site for the insertion of DNA so that we can insert. It should have selectable marker sites also for antibiotic resistance, which facilitates screening of recombinant. And it should possess more multiple cloning sites because we want sufficient cloning sites for the reaction. Coming to classification of vector, depending on the basis of vector, we can classify for bacteria, yeast, animals and plants. Vectors for bacteria which support different kinds of vector, which may be plasmid vector, bacteriophage vector, cosmids, phagemids and plasmids. For yeast, they have special region of replication which autonomously replicating sequence. Example is replicative plasmid vector. And vectors for animal, they should be needed in biotechnology for the synthesis of recombinant protein from genes. Example filament and SB40. Vectors for plants, the production of genetically modified plants has become possible due to successful use of plant vectors. Example TIN plasmid. Types of vector plasmids. So we are going to discuss important vectors which we are using in day-to-day life. The most important is plasmids. These plasmids are small, circular, double-stranded extrachromosomal DNA present in the bacterial cells. They were the first vectors used in the gene cloning. They are found in bacteria, eukaryotes. They are natural, extrachromosomal and self-replicating DNA molecule. They are inherited sharply without the influence of chromosomal DNA. They replicate independently due to the presence of origin of replication. They have high copy number and possess antibiotic resistant gene. They encode proteins which are necessary for their own replication. The plasmids are 1 kilobase pair to 200 kilobase pair in size, which have limited number of genes. The most important feature of plasmid that makes them one of the best vector is the small size. The small size of plasmid facilitate the separation of recombinant DNA from the host genomic DNA. The most bacteria contain more than one copy number of each plasmid. The number of copies of plasmid present in the cell is called as copy number. The copy number of plasmid usually varies from 1 to 50. However, it can be further increased by treating the bacterial culture with chloroamphenicol, which is an inhibitor for protein synthesis. The genes for antibiotic resistance, nitrogen fixation, environmental stresses, etc., occur in plasmid DNA. The antibiotic resistant in plasmids can be used as a genetic marker to identify the strains containing plasmid. Some plasmid also code for secondary metabolites. The small size of vector, however, affects the maximum size of insert DNA it can carry. Plasmid can carry insert DNA. Some plasmid, under certain conditions, integrate into the chromosomal DNA of the bacterium. Such plasmids are called episomes. An integrated plasmid replicates along with the chromosomal DNA. In eukaryotes, except yeast, other organisms do not have plasmid. So the yeast only contain plasmids in the eukaryotes. The example of plasmid is PBR 322. This particular PBR 322 is a plasmid which is of high importance. This is the first widely used purpose-built plasmid vector which is very small in size, up to 4363 base pair, and it is important because transformation efficiency is inversely proportional to the size, and above 10K kilobase pair is very low. Thus, there is a room in PBR 322 for an insert of least base pairs, and they have reasonably high copy number, which is of more advantage. And this PBR 322 is widely used in many cloning techniques. Coming to classification of this plasmid, we can classify based on conjugative transfer and based on function. Based on conjugative plasmids, they are conjugative and non-conjugative. These plasmids which is transferred from one bacterium to other are called as during the process of conjugation are called as conjugative plasmids. So, plasmid which is transferred from one bacterium to other during conjugation. They contain tra genes for conjugative transfer. So, for transferring from one bacterium to other, they are having tra genes. Such plasmids are called as conjugative plasmid, which is capable of moving from one bacterium to other during the process of conjugation. Similarly, non-conjugative plasmids during the process of conjugation, they do not move. They do not move from one bacterium to other. So there is no pass through from one bacterium to other during the process of conjugation. Based on function, F plasmid. F plasmid is a fertility plasmid. Contains some gene expressing the maleness in the bacterium. These genes are also called as stragenes. R plasmids are for resistant. Plasmids contain some genes giving resistance to bacteria against antibiotics and heavy metals. COL plasmid, which can code for synthesis of bacterial toxin, which is colicin. Colicin kills other closely related strains of bacteria. Degradative plasmid. Degradative plasmid code for enzyme that degrade toxic substance such as toluene, xylene, salicylic acid, etc. So, F for fertility plasmid, R resistant plasmid, COL, which is a coding for colicin, degradation for toxic substance such as toluene. So these are the four different plasmids which is classified based on the function. Next coming to cosmid. Cosmids are hybrid vectors composed of plasmid and phage vector. So a combination of phage and plasmid is called as cosmid, which is capable of incorporating 42 kilobase pair of DNA. Cosmid is an artificial plasmid containing cos sites of lambda DNA. It is formed by joining ends of linearized plasmid DNA with the cos sites. It is an artificially derived vector. The cosmid can be a package of capsid of lambda phage in vitro to form recombinant phage particles. It has an origin of replication, selectable marker, gene cloning sites, regulatory sites, and the lack of structural and regulatory genes. Hence, there is no lysis and integration of cosmid DNA in the host cell. No lysis and integration of cosmid DNA inside the host cell. Apart from this, cosmid vectors are large sized vectors with a range of 400 base pair to 30 kilo base pair. They can carry DNA sequence having size ranging from 28 to 46 kb. Cosmid vectors are created in order to incorporate large-sized DNA molecules that cannot be carried by plasmids, since they are the hybrid vectors that can replicate within the host cell like plasmids or remain packaged like a phage. So, plasmids replicate inside the host cell and remain like a phage. Cosmic vector do not have many phage characteristics except a single sequence that promote phage head stuffing. And the example for this particular cosmid is PHC 79. Next coming to bacteriophage. Bacteriophage is the phage plasmid. This phage plasmid or bacteriophage is having higher transformation efficiency, which increase the chance of recovering a clone containing the recombinant DNA segments. It enables incorporation of large DNA under regulatory elements. The use of phage also facilitates the isolation of large quantities of DNA that can be used for analysis of insertion. Even though there are a number of features that can be used as vector, phage lambda is most convenient cloning vector. It can selectively package of chromosome up to 50 kb, and size of phage can be adjusted by removing the central part of the genome, and it is not necessary for replication or packaging of donor DNA. And the example of this is M13. This is a lambda which is having a genetic material DNA which is present with the polyhedral head structure, and tail serves as a attachment of phage in the bacterial surface. This M13 bacteriophage forms a basis of cloning system designed to easily introduce mutations that has been occur in the phage easily. They are circular single-stranded DNA. This genome codes for a total of 10 genes. Next comes the bacterial artificial chromosome. Bacterial artificial chromosomes are similar to E. coli plasmid vectors. So that is similar to plasmids. And this particular E. coli plasmid vectors are obtained from naturally occurring F plasmids, and they are used to study certain genetic disorders and accommodate large DNA sequence without any risk. Next, coming to phage mix. Phage mix are a hybrid vector that has origin of replication from the plasmid and the lambda phage DNA. So this is also a hybrid like cosmid, and this have origin of replication from plasmid molecule and the lambda phage DNA. So combination of lambda phage and the plasmid is phage mix, which is also called as plasmix. It is constructed by inserting a linearized plasmid DNA into a cleaved lambda DNA. This process is generally known as lifting the plasmid. The lambda DNA serves as a site for homologous recombination with the chromosomal DNA of E. coli. Besides this, also helps for in vivo multiplication of phage particles that have recombinant phage mix. They are used in combination with M13 phage. They possess multiple cloning sites and inducible lac gene promoter, which is the advantage for this particular plasmid. They are identified by blue-white screening. These phage proteins are responsible for independent resistance of phage mix. Phage mix can be maintained as plasmid or phage particles in E. coli. The desired gene can be integrated into the chromosomal DNA of E. coli using the phage mix. Plasmid portion can be released free as plasmids from the recombinant phage after introducing it into the E. coli strain. The phage having recombinant phage mix can be stored for a long time. So, these few examples of vectors, and it is very useful for the gene cloning purposes. So the artificial chromosomes, that is bacterial artificial chromosome, or a few other vectors are also there. In artificial chromosomes, we have yeast artificial chromosome. Yeast artificial chromosomes are derived from the cloning vectors used to clone large DNA fragments in the yeast cell. They have linear in shape. Each yeast chromosomes is made up of three important regions: telomeres, centromeres, and autonomously replicating sequence. Similarly, we have huge size comparison with vectors that can be cloned by very large segment of chromosomes, and these particular chromosomes are used in the field of cloning. These chromosomes are sequenced in as a mini chromosomes, and there are two telomeres, one on either side of the pieced chromosome. So many other vectors are also used, and these vectors are highly essential for gene cloning purposes. So the main application of this vector is the vector is a carrier molecule, as we know already, and these vectors are highly essential for the cloning, but they are designed specially for cloning alone, which have certain protein expressions and capable of replicating in the host. So before going for a cloning purpose, we need to properly select a vector, because of the vector only the particular gene replication will happen, and when there is a transfer of DNA from one DNA to another DNA, the vehicle should be in a proper manner so that the replication and expression of gene will happen. So vectors um, are very important for the particular cloning purpose. So in order to get the proper result, proper expression, that is, we need to select the chromosome vectors properly. So for as a conclusion, these cloning vectors are utilized to insert foreign DNA into another cell and create multiple copies of the same. The foreign DNA is duplicated and expressed utilizing the host cell machinery. It amplifies one copy of DNA into multiple copies. So selection of vectors is very important in the gene cloning technique. Thank you.